So this is Project 2, Chapter 10, with the learning objective, describe the biological basis of impulse control, including neurochemicals and neural structures. So first, we're going to look at impulsive behavior through the lens of serotonin. So aggressive behaviors are species typical and are regulated by neural circuits in the brainstem, specifically the amygdala and hypothalamus. So how does serotonin come into play? So activity of serotonergic synapses inhibits aggression and other risk-taking behaviors, while the destruction of serotonergic axons in the forebrain facilitates aggressive behavior. So there's many studies that have illustrated this, but I found the one about the free-ranging colony of rhesus monkeys, which was observing the relationship between serotonergic activity and aggressiveness to be the most helpful. So the researchers removed a sample of cerebrospinal fluid and analyzed it for 5-HIAA, which is a metabolite of serotonin. When serotonin is released, most of the neurotransmitter is taken back into terminal buttons through means of reuptake, but some does escape and it is broken down into 5-HIAA, which travels down to the cerebrospinal fluid. Because of this, high levels of 5-HIAA in the cerebrospinal fluid indicate an elevated level of serotonergic activity. So the research found that young male monkeys with the lowest levels of 5-HIAA showed a pattern of risk-taking behaviors, impulsive behavior, aggression, specifically towards monkeys that were older and larger than them. And a large percentage of the monkeys with the lowest 5-HIAA levels died, and this was because they were fighting much more experienced monkeys, older, larger. If they were doing a fight, they could not win, and it was because of these low levels of 5-HIAA. So why does it matter if monkeys with lower levels of 5-HIAA engage in aggressive, risky behavior? So it looks like studies with humans have found that this is the same case for humans. So serotonergic neurons play an inhibitory role in human impulsive behavior. Specifically, low rates of serotonin release, represented by, again, low levels of 5-HIAA in the cerebrospinal fluid, are associated with aggression and other forms of antisocial behavior that are innately human, such as assault, arson, murder, and child beating. It makes sense that substances that are serotonin agonists would reduce these type of behaviors. So serotonin plays a huge role in impulse control, but so does the ventromedial prefrontal cortex, which inhibits responses and plays a role in emotional regulation. So the ventromedial prefrontal cortex receives signals from the dorsal medial thalamus, temporal cortex, ventral tegmental area, olfactory system, and amygdala. It then communicates with the cingulate cortex, hippocampal formation, temporal cortex, lateral hypothalamus, amygdala, and other regions of the prefrontal cortex. Because of its inputs and the information it's receiving, the ventromedial prefrontal cortex is provided with information about what's happening in the outside world, while its outputs allow it to impact behavior and physiological responses, including emotional ones. So the most obvious and fundamental way of illustrating the importance of the ventromedial prefrontal cortex in impulsive behavior is then to look at the effects of damage to this region. So patients with lesions in the ventromedial prefrontal cortex act impulsively and enact sudden bursts of anger. Because they lack emotional responses in important situations, they also make poor decisions. The prefrontal cortex plays a role in moral judgments. So when someone has to make a decision between a utilitarian judgment versus a personal moral judgment, the ventromedial prefrontal cortex is activated. So here I'm going to present the trolley dilemma. Some of you may be familiar with it. I know I was. So you witness a runaway trolley with 5 passengers heading towards a cliff. You have access to a switch that will cause the trolley to switch tracks, saving the life of 5 people. However, a worker is standing on the other track and he will be killed if you hit that switch. In this scenario, you are making a utilitarian judgment because even though 1 person dies, 5 live. Now consider the same scenario but with no switch. You are now standing on a bridge above the tracks along with an obese man. Pushing the obese man would cause him to fall on the tracks, stopping the trolley and once again saving the 5 people. This is a much more difficult decision. This is now a personal moral judgment, as you are physically pushing a human being to his death. Surprisingly though, people with surprisingly or not surprisingly, I suppose, people with damage to their ventral medial prefrontal cortex display much more utilitarian judgments. The regular person would agree with the first scenario and doing the switch, but actually these people with damage and lesions are more willing to push the man in the second presentation of the trolley dilemma. So these come together, and the roles of serotonin and the ventral medial prefrontal cortex to play a role in impulse control. This is shown by 9 psychopathic people being reluctant to harm others because just the thought causes very negative and unpleasant emotions. The release of serotonin in the prefrontal cortex activates this region, and the serotonergic input to this region is responsible for the ability to inhibit aggression and risky behavior. A single high dose of serotonin agonist decreases the chances of an individual making utilitarian decisions in moral dilemma tasks. Therefore, depressed rates or low rates of serotonin in the ventral medial prefrontal cortex are associated with impulsive and aggressive behavior. So I found this article that looked at aggression in mentally ill offenders and the role of 5-HIAA. I thought it was interesting to tie it since we did mention the importance of the metabolite in rhesus monkeys. So Lidberg in 2000 aimed to study the relationship between low levels of 5-HIAA in the cerebrospinal fluid and aggressive behavior in violent offenders. So they measured levels of 5-HIAA in the cerebrospinal fluid of 35 drug-free men who had been convicted of homicide and who were stationed at the time of the experiment at a psychiatric hospital undergoing evaluation. The research found that the mean concentrations of the metabolite in the homicide offenders were no different from the control subjects. However, they did find that 1/3 of the offenders had attempted suicide and that their mean concentration of the metabolite were much lower than that of the other offenders. Additionally, participants diagnosed with impulse control disorder also had lower averages of 5-HIAA. Again, I found this study interesting because as we saw in the study of the rhesus monkeys, low levels of 5-HIAA in the CFS were correlated with riskier behavior and more aggression. Suicide is definitely an impulsive behavior, and it is no surprise that the subject who had attempted it had the lowest average of 5-HIAA in their cerebrospinal fluid. This study just goes to show the importance of serotonin and its metabolite in impulse control and behavior.